Hi everyone. Today I'm presenting about my image, which is titled Periodontal Petals. And this is an incredibly collaborative work between three different labs at MIT Chemistry, MIT IMES, and the VIDMC in Harvard. So we're studying periodontitis, which is a chronic inflammatory disease that happens in the oral cavity, particularly in the gums. And in this disease, the gums are like inflamed chronically. And this is a disease that affects almost 1 out of 2 adults globally. So on the left, you can see that this is like how your natural gums look like, where your bacterial biofilms are sort of outside the inner connective tissue of your gum. But when you have periodontitis, you have sort of breach in this barrier of your gums, and the microbial like species can sort of invade into the inner connective tissues that causes inflammation and loss of like the tissues that sort of support your tooth or your dental implant that leads to loss of tooth in general. And this is a very difficult disease in the sense that there is no actual cure for periodontitis. And the only way to like sort of like maintain this disease is through maintaining proper dental hygiene, but it is only manageable and there is no like sort of therapy against periodontitis. And the reason periodontitis is incredibly alarming is that in recent years, it has been shown that periodontitis is associated with a lot of like different systemic comorbidities. It is associated with diseases even in the brain, like Alzheimer's disease. It is associated with different sorts of cancers in the GI tract. It is also associated with diseases like myocarditis or colitis or obesity. And this makes it important to study this disease and find out some cures that can be used for periodontitis. The way we sort of approached the problem was to understand how the different cell types that are in the gum, how are they affected individually when you have this sort of inflammation. And the way we did it was by using spatial proteomics. And we used a method called Codex, where we have a sort of different DNA barcoded antibodies that can target different proteins that are present on different cells. And we sort of pool these barcoded antibodies together and stain it on the tissue. And then by looking at each of these barcodes, we can get these incredibly multiplexed images that tells us where different proteins are in the tissue on which kind of cells, and helps us understand like what is happening at a single cell resolution in the tissue. And this has been like one of the methods of the year for Nature Methods in 2024. And what this kind of spatial proteomics does is that it gives these incredibly multiplex images that gives us a like a way to like deep dive into the tissue and what is happening there. So here is our image where we are staining for different kind of protein markers. But two things that I would like to show is that these green areas that you see are your epithelial layers, whereas like all the other blue regions are basically your fibroblast layers. And generally, one of the things that is very hypothesized to be very important in the periodontal tissue are these stem-like epithelial cells. And we can stain for these stem-like epithelial cells, which are these neon-colored like patches that we see in the epithelium. And one of the things that is incredibly stark and like interesting to us is that these neon patches always appear in regions around fibroblast niches. And when the epithelium is like far away from the fibroblast regions, it becomes more differentiated. So you do not see these neon patches anymore. And these like stem-like epithelial cells have been known to play important roles in inflammation, but also in other contexts like oral cancer. So it gives us an incredibly like powerful way to like understand what kind of biology this fibroblast stem-like epithelial crosstalk sort of plays, and like how they help maintain tissue homeostasis. We also focused a lot on the immune cells because we wanted to understand like what is driving this inflammation. And one of the things that we stained for was in this like blue color, which is like the CD3, which is a marker for T cells. And what we find is that in these periodontitis cases where you have like, this is like a stage 4 periodontitis, which is like a very aggressive disease, you have a lot of infiltration of these T cells into regions that are mostly stromal, like epithelial or like fibroblast regions, which is not seen in healthy tissues. And this kind of gives us an idea that there is some sort of like deep biology that is happening that is making these immune cells sort of infiltrate into normal stromal regions that is probably driving the inflammation. And to look into it more, we stained for this marker in green called ICOS, which is a sort of marker for inflamed like T cells. And we see that like there are a lot of these T cells that are infiltrating into the stromal layers are expressing this ICOS marker. So this gives us an idea that these T cells are probably driving and contributing to this inflammation that happens in periodontitis. And if we can sort of like target these T cells, then we might be able to target this inflammation and get a cure for periodontitis. So I would like to thank all the different people in the different labs, in the Sherlock Lab, and the Harvard Dental Center, Harvard School of Dental Medicine, the Jiang Lab, and the DFCI Core, who have contributed to this incredible project. And we look forward to finding new biology that can help us find a cure for this disease. Thank you.